for a while. And now he's back at Mizzou. But what I didn't maybe appreciate was that he's quite the world traveler. So he did high school in Nepal, his bachelor's in Bangladesh, and his PhD in Japan before coming to the US and doing a postdoc first with Mike Skinner at Washington University in Pullman, and then at Mizzou in Columbia, Missouri, before taking his first faculty position at UNC Greensboro, and then moving to Mizzou just fairly recently. Yeah. So now he's an associate professor in the Division of Biological Sciences at University of Missouri Columbia, a reproductive biologist by education, environmental health scientist, epigeneticist by training. It's been quite successful, I think. So he's published 3 book chapters, more than 75 peer-reviewed manuscripts. He has several NIH grants and really funding from NIH, USGS, University of Missouri, University of North Carolina, lots of different funding sources. Is very involved with service. He's running a meeting in Portugal just shortly. And overall, just a terrific collaborator. We learned together early on the value of good collaborations with some not so great ones, but got a highly cited co-author publication out of that. First author for Ramji, whether or not we would collaborate with those people going forward, but at any rate, very excited to have Ramji here, and I'll let him. Thank you.
Yeah, thanks, Chris. It was a wonderful introduction, and it's really nice to come back here and to be invited by you and the Wayne State University. So it was also really nice to see you growing to this level. I have seen him since a grad student, and then I have seen him as a faculty. And meeting his lab, it feels like, wow, time is flying so fast. Okay, so thank you for coming after lunch here to listen to my talk. I'll be talking about there are some, and if we can make some efforts to mitigate environmental health-induced transgenerational health outcomes through epigenetic reprogramming of DNA epimutations. It's pretty like heavily loaded title, but in short, what I want to say is environmental chemicals are everywhere. In our diet, in our in the air, in our water, and we're exposed to them every day. And the chemicals have been really integral part of our life. We cannot survive without them because industries make their product for our comfort, we demand them, and then we also go against them. You made them so much, and we do not want them anymore, but we still use them, right? So it means it's a never-ending story. So we need to find out a way to actually protect human health or organismal health, and we have to go through a different route to find out if we can develop a secondary, process of helping organisms and humans so that we can at least either block the disease or cure the disease, or even do not let these chemicals enter our body. Okay. So with this, first of all, I would like to thank Wayne State University, particularly the P30 Center, for inviting me. And then, of course, Chris, everywhere in a conference I go, I see him. I feel proud to meet with him in his lab. And then I also like to thank the funding agencies. Without their help, I would not be able to come here today. And also would like to thank my collaborators, my previous mentors, who constantly actually inspired me to be in this field and helped me through professional development training, as well as helping me how to write grants and giving me guidance in every step when I was in the process of transition from postdoc to faculty. And I also would like to thank some of the folks in U.S. Geological Survey. US Geological Survey over here, where I actually learned a lot of things that happen in the government and also learned some techniques and my colleagues in well research across the globe. So, and this is my lab. Without their contribution to research, I would not be here today either. So the second thing is I'm also looking for some people, if you are interested, for a technician position who can do epigenetics, or I need 2 graduate students and 2 postdoctoral fellows, especially on AI-based big data analysis to determine the pathway to disease as well as the biomarker to disease. And my research is completely based on the concept of gene-environment interaction. If you look at this figure over here, you can see in early development, genetic processes, which are the foundation of life, they actually determine the very precise gene expression pattern. Their disruption can lead to alteration in the development as well as physiological changes later in life. So there is another process called epigenetics. This is, this process also works similarly, but it acts on DNA to alter the expression pattern of genes and making those similar physiological changes that are associated with health and disease. But here comes environment and diet. So the environment, for example, I have listed some of these components over here. They are part of the environment. And they can induce some genetic changes that can cause DNA mutation sometimes, but in majority, they are able to change, make epigenetic changes. They can alter these differential gene expression pattern or like chromatin organization, leading to physiological changes later in life. These changes happen in early development, and they can be carried over to adulthood, and sometimes these physiological changes are associated with adverse health outcomes. Those outcomes can be beneficial or harmful depending on what type of outcome they are. And a lot of times we have seen these outcomes are able to travel from parents to children to grandchildren without making any DNA mutation across transmission process. So I'm interested in this part of science. So how these environmental chemicals establish the changes in the individuals in early development, and they result in the phenotypic outcomes in the subsequent generation. So the question I usually put these questions for my students when I teach in the classroom, and students usually try to ask these simple questions about what's going on, because every time the human health is the topmost priority for everyone. So the known question is humans, what is known so far is humans want to live a comfortable life, right? We want comfort. So whatever way we can achieve that comfort, we are ready to do that. So the industries make those products for us. And then what happens is those industries are making all the products that we use and they're component of our life. And we're living that life constantly. But the thing is, we do not know, right, these exposures or diet that we take are inducing the adverse health outcomes that we have today, or that adverse health outcomes we have today without exposure, but because of our parental and grandparental lifestyle, at that time they were exposed. We do not know, right? So the second, we do not know a lot, but our literature shows that this is happening. Right? The third one is many chemicals exhibit the non-linear dose response. So it means very low concentration chemicals are also making the same type of change as very high dose chemical. And these effects can be heritable as well. Those low dose effects have been found to travel through the germline to their offspring. And then unexposed offspring of the exposed parents can still develop the disease due to epigenetic inheritance of those memories. So that has been shown experimentally in animal model. And then also a lot of epidemiological studies have also found the association of those changes in the human population. They have started coming up slowly. But what is unknown here, right? We know all of them because of the research that we did for the past like 40, 50 years. But what is unknown is, can humans and non-human organisms be protected from environmental chemical exposure induced adverse health outcomes? Can we actually protect? Second is, can environmentally induced heritable traits be mitigated? Means, can we reverse them or can we get rid of them after they hit our body? And then the third one is, can future generations be protected from the environmental endocrine disruption? So it means if we make the environment clean, are these future generations still susceptible to endocrine disruption? Or can we make our future generations completely safe from endocrine disruption of the upcoming chemicals that will be there 20 years later? Okay. These are the questions we do not know right now. So we need to do some study about that. And my laboratory and others actually showed, I'm giving you an example over here, how we developed our hypothesis. This is here developmental exposure using fish as a model. You see those red stars means they are phenotypic defects in the liver and gonad. So developmental exposure to BPA, we showed in 2015 that causes fertility defect in same generation and then subsequent generation and grandchildren generation. It goes through 3 generations down the line. And then ethylene does the same thing. And if we use atrazine, does the same thing. And then methyl mercury, somebody else published using the same model, same window of development. The methyl mercury gives rise to the fertility defect during that window of development. And THC, which is like cannabinoid, which is a different chemical, still produces the same result. So it means like what is happening here is exposure is occurring at this time of window, which is 0 to 15 days post-fertilization in early embryogenesis. No matter what chemical they are producing fertility defect in the later adulthood, and then you can see them in the grandchildren and children. Okay, so the idea is, do these chemicals have a common mechanism of action in developing germ cells, right? Regardless of that. And then the second is, one thing you can notice over here, the chemicals can have a different mode of action maybe, but however, when the disease develops, they all funnel into a very narrow pathway, and that pathway leads to fertility defect. Okay. So if we look at more closer, it means gene-environment interaction during the same developmental period here induced these two defects, like fertility defect as well as fatty liver disease, that we recently noticed when we went back to do histological examination of all the samples. We found out not only fertility defect, we found out the fatty liver disease as well. And that was more pronounced than fertility defect, suggesting that at this time of development in germline cells, something is going on. Any chemical that goes into the body of these embryos is making that permanent signature that is giving rise to these fertility defects later in life. So that is one pretty cool observation that we have. And then we developed as a group of scientists, we developed a kind of hypothesis and wanted to ask questions about how the germ cells memorize life-related experiences like diet exposure or environmental stress. How do they translate these exposure memories into the physiological signal, leading to the phenotypic trait with compromised health or fitness in the subsequent generation? Can these memories be reprogrammed and health be restored? So that is the question we asked as a group. So to answer these questions, we use basic biology concept, particularly cell biology, development, epigenetics, genetics, and physiology, biomedical toxicological concept as well. And then we use computational biology and artificial intelligence to run these big data sets. So collectively, we are aiming to answer the questions in environmental health, environmental, human, and ecosystem health. So we use bisphenol A as a chemical of interest because we have found the bisphenol A, in the literature that bisphenol A is everywhere in the world, and then the people who have been, whose blood and urine has been measured so far have some certain amount of bisphenol A in their circulation. So it means there is occupational exposure as well as the normal environmental exposure going on in humans and non-human organisms. Second is we're using 3D in vitro culture method 2D and 3D organoids. We're using rodents as needed, and then medaka fish most of the time as animal model. And then we take epigenetic mechanisms as drivers of those developmentally established changes that are turning into disease. So why medaka fish? Medaka fish is a very small fish and smaller than zebrafish, and it can produce sperm and egg as many as you need. There is unlimited everyday spawning. You can collect them. And then also the embryos are transparent. You can actually manipulate them outside the body, right? And other similarity with mammal is their germ cell development is similar to mice. They have genetic sex determination, XXXY, and medaka package the sperm genome just like mammals with protamine. And the other spectacular observation is that we saw the epigenome reprogramming process is conserved between mice and medaka, especially in the germline cells, suggesting that if we want to study the inheritance through germline, and there is an overlap that we can use and still make some very useful information out of it. So this is the DNA methylation pattern. For example, here there are two phases of reprogramming, and in male and female show similar pattern like mice. So the dotted lines are mice, and then here the orange line is medaka. So taking this information, we are putting their reprogramming mechanism into our table to understand further. And then why epigenetic mechanisms? Because epigenetic mechanisms as drivers is our way to go, because gene environment interaction leads to a very little genetic mutations which are permanent, whereas the majority of them are epigenetic alterations which can be reprogrammed, erased, and reestablished during the lifetime of the individual, suggesting that there is a room for correcting the epigenetic changes that are established by the environment. So to do that, we use medaka fish and we expose to BPA. So we use BPA 10 microgram per liter, and for 15 days post-fertilization, same window that I showed earlier, and then never after that. So after 15 days, they were never exposed to BPA. Okay? So then we measured how much BPA is taken by eggs, which is 28 picogram in 1 day. And then the medaka eggs are about 1.2 milligram inside. So suggesting that the amount of BPA they are taking is very, very small compared to the dosing concentration that is in the environment. And then we looked at the F2 generation, because we already know that BPA can affect the fertility and liver health because of direct exposure to either these individuals or their progeny. But we did not know in the past that it affects or not the subsequent generation. So we repeated the experiment with lower and lower concentration, but this is the concentration we chose because it is found in many parts of the world. And then also the uptake we actually determined using this concentration. So what we did is we did histological examination, and then also used the endpoints like liver histology, gene expression bulk and single cell, attack-seq, and epigenetic alterations like bulk and single cell methyl-seq, metabolomics untargeted, and protein expression untargeted, and then microbial dysbiosis. We have not done protein expression yet, but we have the sample for that. However, we have done all others. I'm presenting only those that are highlighted as orange today. So the phenotype, what we got, if you look at, because of early embryonic exposure at F0 generation, you can see reduction in fertility of the F0 generation in the BP lineage, right? Here in F1 generation, they still keep that lower fertility, and in F2 generation, it's still low. So suggesting that the fertility defect is traveling across generation. So in addition to that, what we found is from F3 generation onwards, so the BPL lineage individuals started showing behavioral problem, right? So they started chasing same-sex individuals before they find a partner. It took about 3 hours for them to find a right partner, a female found male, a male found female, to mate with. By that time, the controls already mated and produced their eggs. Okay? So we also saw that there was a repeated somersault-like behavior in the BPA exposed lineage from F3 to F7 generation continuously. So then we consulted with some neurobiologists, and they said there might be neurological disorder going on in them. So they are now trying to see whether this is kind of typical neurological behavior specific pattern or something else. We are now trying to go back to the video and reanalyze again. So if you look at the testes of these individuals, the F2 generation, so you can see over here that their seminiferous tubule is broken. Most of the cells are in sperm, and then most of them have lost the tail. Whereas in the control, you can see over here that control has the pretty good layer of germ cells, different stages, and then also the seminiferous tubules are intact, suggesting that this fertility is massive for males. And then here, my grad student at that time, now he's a postdoc, he actually wanted to look at the ovary. He found out those individuals, F2 generation females, had smaller ovaries, right? And then compared to control, and he looked at fecundity, egg production, and fertilization rate, both were significantly decreased. When he looked at the histological figures, he found out control lineage has a very nice vitellogenic, oocytes that are found in normal, adult female. But in the BPA treated females, they were arrested in the early stage. And he counted them, found out there was significant increase in number of the early stage oocytes, whereas the late stage oocytes were completely decreased, which is the kind of indication of having polycystic ovary syndrome in humans. So then he published that paper and found, he declared using the gene expression markers, further clarifying that these are the markers that are expressed only in the polycystic ovary of the adult human patient. And he also did some metabolomic analysis to further confirm that. So it means that these individuals, the F2 generation females, were developing the polycystic ovary-like phenotype in the grandchildren generation because of grandparental BPA exposure. And then another student did some microbiota analysis of the same individual that had the fertility defect. So this master student, Alyssa, actually did some 16S, quick 16S analysis and found out that the Cetobacterium, which is poor microbiota, microbiota is decreased in the BPA lineage, whereas the pathogenic bacteria like Pseudomonas, they were increased in the gut of the female fish. In the males, there were some other different type of microbiota, but since the female had the phenotype, I'm presenting the female result here. So what she found additionally is this early grandparental BPA exposure caused dysbiosis of the gut microbiota in grandchildren. And then she also found that there was altered intestinal epithelial epigenome and transcriptome profile in those individuals. And then those intestines were extremely fragile. She was lifting with tweezers and they started breaking apart. That means they were so thin and there was leaky gut syndrome going on and they were not feeling well, not digesting the food very well. And then additionally, the pathogenic bacteria colonized the gut of the BPA lineage fish in the F2 generation. And then he wanted to see what other phenotypes are there, and he looked at the liver. And if you look at here, the BPA lineage fish were a little fatter than the control fish. And he found out the liver was white. So suggesting that the liver is undergoing some sort of fat accumulation compared to the control liver here, which is kind of more pretty normal. And he did histology and found out in males, the severity of liver disease was a little bit lower than the females. Female had accumulated a lot of fat content, fat in the liver in the F2 generation. And then he looked at the hepatosomatic index, the liver weight, compared to the body weight. He found out the hepatosomatic index was increased in the BPA lineage as well as in the female. And then he also looked at the vacuolated cells, means the cells that had nucleus moved to the edge and which looked like a vacuole, and they were increased, and 3 out of 6 males had it, and in the female, all of those females had the vacuolated hepatocytes, suggesting that it was a clear indication of the non-alcoholic fatty liver disease. Then he did some, RNA-seq analysis and found out the lipogenic genes were, increased and lipolytic genes were decreased. Lipotransporter, lipid transport genes were also increased, and apoptotic genes were increased. So, which is a typical signature of the liver disease. Then he looked at in the F4 generation further to see whether this phenotype continued, and found out that F4 generation still maintained the fatty liver phenotype, and additionally the those individuals actually accumulated macrophages and also underwent the fibrosis stage. So means which is the indication of development of steatosis or fibrosis in the liver. And when he actually counted those cells, live versus dead, found out that cells were dying. 9 out of 10 fish had the liver cells dying off. And he looked at the gene expression pattern of some of the markers. The PPAR alpha, which is lipogenic, was lipolytic, which is decreased, and then SCD, which is lipogenic, is increased. So he wanted to see, okay, it is going on. Let's go back to look at the sperm of the father, grandfather, father, and then their descendants. So what he did, he collected the sperm of F0 generation founder, that were directly exposed to BPA during early development. And he looked at the F1 sperm, and then he looked at the epigenome of the liver somatic cells, right, the hepatocytes, as well as he also looked at the gene expression pattern in those hepatocytes to see whether there is a direct link. And then he did WGBS, whole genome bisulfite sequencing, and coverage was about a little bit lower than we expected. We usually do 60x, and 60 million reads, and almost like 30x. But he focused only on the promoter because he wanted to make a canonical connection with gene expression, right? So he did whole genome analysis, but I'm presenting the data from only promoter part of it. So now he's a postdoc with me, working with me for about 2 years, looking for faculty position. If you are trying to hire someone, he's the person who rocks, actually. He's a really, really great guy. And then he has almost 12 publications, from his PhD to postdoc transition. And then if you look at here, F0 sperm had the DNA methylation on the left side, which was hypomethylated in the sperm. If you look at the F1 sperm, it was half hypomethylated, half were like hyper. And in the liver, when it was transmitted from germline to soma, liver maintained completely hypermethylated genome, both male and female. So it means that the genes we expect probably are shut down in the liver, right? And he did this Venn diagram. If you look at here, comparison, how many genes, how many promoters were represented. And here is 1156 promoters were indicating, F0 sperm was indicating the F2 liver will have fatty liver disease. They were all specific to fatty liver disease. And when he put this data into more specified and targeted analysis, he found out that those genes that are involved in oxidative stress, they were increased, and the genes that are involved in lipid metabolism, they were decreased significantly. And if you look at those genes, if they are parallel to human NAFLD patients, he pulled out those genes and then compared with the human database from this Science Translational Medicine manuscript that was published recently. So he found out most of the genes that were found to be upregulated were actually found in the medaka fish as well. So it means human NAFLD patient specific biomarker of that disease were found in medaka fish clearly, suggesting that the medaka fish is developing this NAFLD phenotype, which is similar to humans molecularly and also histologically. Then he wanted to see whether it is what type of disease, and we found out, looking at the histology, by 60 days there was no sign of liver disease onset. However, by 90 days, which is 3-month-old fish, they started showing the lipid accumulation in the liver. So then he treated them with AJA, which is DNMT inhibitor, just to see, because there were DNA hypermethylation in the genome. the genome-wide methylation was like, shifted towards the right, and almost the genome was hypermethylated cumulatively. So what happened after treating with AZA, like there was inflammation in the liver, and fish did not eat, they lost weight, and water accumulation happened in the intestine. The fish were suffering from that pharmacological treatment. And then he found out a Nature paper that talks about vitamin C can be a weak epigenetic modifier. Then he used vitamin C to treat them from 60 days to 90 days at the same time period, before the onset and towards the development of disease. What he found that he only was able to, the oil droplet size was decreased and cell death did not change with that time period. Then he wanted to go little forward and earlier, at the time of day 30, which is almost the time for pubertal development in fish. At that time, the liver is more like liver molecular signatures starts more establishing more like a what can I say, age-related or sexual development-related changes happen in the liver at that time. So he wanted to target that time point. If you look at here, this is BPA treated lineage F4 generation, and then he treated with vitamin C, and it was completely cured, which is almost similar to the control. And he looked at the DNA methylation profile and RNA-seq result to see whether this correction actually involved the correction of those DNA methylation patterns that were established. So what he did is, if you look at here, these are the chromosomal pictures. And here in the left-hand side, the arm is BPA for that chromosome, and right-hand side is vitamin C in the F4 generation. He used 100 BP tiling, and then the 20% methyl, the criteria was 20% must be methylated, and then FDR of at least 0.1, less than 0.1. So you can see over here, these are the red color are hypermethylated. In the the left arm is, if it is hypermethylated, it is red. In the right arm, if it is blue, then it is corrected by vitamin C. If you look at that, that small picture over here, you can see these three were completely corrected, and those two were completely corrected. And this way, we screen for those that are corrected. There were so many of them, they were not corrected, still showing the same pattern. But however, we only selected those that were corrected here. So what you see over here further is, you see all of those, most of the changes that are accumulated in the ages as well, like telomere region. So suggesting that probably there is a liver aging going on, epigenetic aging of the liver, and vitamin C, most of them, in most cases, the vitamin C corrected them. Suggesting that probably some of the aging related genes are also being corrected by the vitamin C treatment. And he looked at repeat elements like transposable elements, because among these genes that were corrected by vitamin C, we wanted to see the nature of those. Like, then we found out some of them were actually represented the repeat elements or transposable elements of these kinds. So suggesting that probably there is a kind of massive reprogramming going on at the epigenetic level, genome-wide, by vitamin C treatment at that time when the liver is about to transform into an fully adult liver. Then we did some specific changes, right? These are promoters. We wanted to see whether these promoter changes are like very, very specifically changed. So here, if you see in the F4 generation, there's the left-hand side is hypermethylated promoters. For example, these are the genes that are associated with fatty liver disease in humans. And then here you can see corresponding changes. So those color actually show in a slide, this one is vitamin C treated on the right, and the left-hand side is control. If you look at here in this middle, so these are hypermethylated promoters. They were completely reversed by vitamin C treatment among this group of genes that are very specific to human NAFLD patients. And then if you look at others, they were slightly corrected, but vitamin C corrected more. And they were more corrected here in the control. They were normally in control, but in vitamin C only slightly corrected them. Suggesting that there is correction, but there's a different degree of correction going on. And if you look at here, particularly on the right-hand side, these are the expression pattern of the gene. So these changes corresponded with the expression pattern, which are down regulation of the target gene, the same gene that has a promoter hypermethylation, were actually down regulated. And then with vitamin C treatment, they came out. So these fatty liver genes came up like to normal, so the phenotype was corrected, right? So then we also did one quick experiment in human cells, right? We grew the human cells. These are THLE cell line. They are normal human liver cells. And we put 50 micromolar BPA for 48 hours. And then fat accumulation started showing up. And then when we use vitamin C, the fat accumulation decreased. But interestingly, when we put the combination of vitamins like A, B, C, and E, it did not make any change. So suggesting that the vitamin C is specifically working on the liver to actually alleviate the fat accumulation process in the liver in vitro as well. And there are here are some data that are not specific to human NAFLD patients, but we saw significant change in our database that these are the genes that were down-regulated in the BPA lineage, and because of vitamin C, they were upregulated and they were comparable to control. And there are the genes that were upregulated by BPA, exposure, grandparental, but because of vitamin C, they were down-regulated and they were comparable to control. But they were not the genes that we found in the human NAFLD database, but they are medaka specific changes. But interestingly, we also found mitochondrial changes, right? We actually separated some gene expression differences. You can see over here in the left-hand side, the BPA individuals, all like mitochondrial genes that we looked at, they were completely down-regulated in the liver, and with in control, they were they show this pattern, those two, and vitamin C actually reversed them, and even did much better than control themselves. So vitamin C potentiated the liver health even by improving the expression pattern of mitochondria in the liver. And we also checked for the aging-related genes in the database, and there were 25 aging-related genes found out that are specific to human aging. So 10 out of them, 10 out of 25 were restored by vitamin C treatment. And then if you look at other, just the promoter versus gene expression, other genes that are not related to NAFLD, you can see the DNA methylation over here. The red is completely reversed into blue with vitamin C. Their gene expression is also reversed here on the right-hand side. If you look at to the next one, over here, so this is more canonical at the promoter region. So some of the promoters that were highly methylated, and they also have nothing to do with NAFLD disease in humans, but still they are found in medaka and zebrafish to be associated with liver disease. And here you can see the control, which is maintaining normal promoter methylation. And then you can see here the promoter hypomethylation because of vitamin C, suggesting that not only those the human specific genes, the genes that are involved in liver, maintenance of liver in medaka, are globally reversed by vitamin C. Not only that, we had this, polycystic ovary syndrome phenotype, right? So when we treated the same individual with vitamin C, right, when we looked at the histology, we found out that the same fish, same exposure, time point. So what we found is vitamin C corrected them back to normal. So the polycystic ovary syndrome also came back to normal, suggesting that probably there is a systemic level change going on in the body because of just kicking off the change in the liver. So now he is analyzing what are the genes that are corrected in the polycystic ovary syndrome with vitamin C, and also he's also trying to find out the epigenetic alterations. Now this week we actually sent the gut microbiota sample from F4 generation to sequence whether the dysbiosis was also corrected, because the intestinal lumen, also the epithelium started showing up a little bit of opaque color, which is like little thicker than the untreated ones. So then the last part here is since we know right now the vitamin C actually reversed and reprogrammed the epigenome back to comparable to control. Now we are developing a CRISPR-Cas9, dead Cas9 method to target epigenome. So this is the gene that we found in our database from F0 to F4 generation. It constantly showed up to be overexpressed, and then its CPG island here in the core promoter was hypomethylated. So we wanted to bring this hypomethylation to hyper and then shut down this gene, because this gene is involved in the humans. This is this starts expressing at the stage of NASH, and it shows up really high in the liver cancer patients. So suggesting that this gene is the enzyme that actually produces bile, that also involved in proline biosynthesis and arginine. In our metabolomic analysis, we saw this enzyme come up, as well as in our metabolites, we saw proline arginine pathway being activated. So clearly shows that this gene can be a really good target to start with. And we are now doing is we are injecting with CRISPR dead Cas9 DNMT3B into the fertilized egg of F4 embryos. These are the F4, they have embryos, and their F5 has fatty liver disease already. And then we have, we used one guide RNA because it can cover all 16 CPG sites within that small window of sequence. And then we are now checking the phenotype, because phenotype development takes time. Right, 90 days to show the phenotype. We recently injected it. Now we are checking whether it will develop that phenotype. We are also doing this targeted sequencing of the liver cells to see whether these changes can be reversed. And then also we are injecting, my student just last week, he injected into the adult liver, F4 generation fish, directly into the liver to see whether these CRISPR constructs can get into the neighboring cells of the injected site, so they can, whether that actually can make any change without having any cell proliferation in the differentiated tissue. So the last one slide, that interestingly using the AI that my bioinformatic person is developing, putting all our data together. Like, gut microbiota, liver disease, reproductive disease. What he did is in F0 generation, we had mild phenotype of reproduction as well as, liver disease. We had 15 to 20 only. And then he put this together and found out there was no correlation because of the 50% disease between ovary and liver. Those epigenetic changes, epimutations did not have any, overlap, and they did not segregate. And then in the F1 generation, it still had less than 50% disease, and they started showing a little bit of segregation, but still it was not statistically significant. Then in the F2 generation, they clearly segregated. So then using the epimutations in the sperm, right? So you will be able to see these individuals have the ovary or liver disease. So these signatures are indicating they have fatty liver disease, and these signatures are indicating that they have the ovary disease in these individuals. So suggesting that probably we will be able to use this type of analysis in the future, because it is a very small sample size. But if we go to the clinic and find out buccal swab, right, if they have the transgenerational history of exposure in the past, the buccal swab can serve as a sample. We can do some whole genome analysis of the samples to find out those segregated markers that can probably predict the disease those people have. So we are trying to develop this strategy together with medical school community in our university. And with all this, I would like to conclude here with this summary that we saw the fatty liver disease because of ancestral exposure, and those memories were traveling across generations. And then we have to have, with a simple epigenetic modifier like vitamin C, we can mitigate the exposure-specific liver disease, but we have to target them at the time of very critical window of susceptibility to that particular modifier. And then the reversal of phenotypic trait was associated with correction of transgenerational epimutations, and then reestablishment of pathway that for healthy liver function. So So with all this, I would like to thank you for your attention, as well as I'll take any questions you have, and I always put this slide at the end.
Great talk. Fascinating work. So two questions. One, maybe I missed it, but the vitamin C concentration that you're using, how does that translate into human intake? So is it just one vitamin C capsule a day? Is it less than that? Do you have a way to translate that into human vitamin C exposure?
We are not there yet in terms of comparing the concentration, but we use 1 milligram per milliliter.
Okay.
1 milligram per liter in water, because it's water soluble. And we have not tested the uptake by the fish, and we need to have some kind of radioactive
Marker,
marker to do that. And I think that is the next step, because this is all preliminary data, right? So we're now developing it into the full-fledged, research proposal to NIH. It got like 9%, I didn't get funded. So anyway, no, 13%, I'm sorry, no, 9 was the other one. So 13% I didn't get funded. And of course it was not 13%, but it's still, I was hoping. And then the other thing is probably, if you look at 1 milligram per liter, most probably is already human relevant, because people use 1,000 milligram, right, for over-the-counter vitamin C, and from 400 to 1,000 milligram every day, because it's water soluble, and then also it needs iron to be, to digest it, right, transport it from the intestine. And in circulation in human blood, probably, I'm not sure, I think 80 nanogram of vitamin C can be seen in circulation, and which might be closer to what we have, but we need to actually test.
Yeah. So the other question, follow up on the vitamin C, is I was really interested in the mitochondrial upregulation of the mitochondrial genes with vitamin C, but at least in humans, I don't know how it is. In fish, mitochondrial genome is not methylated. So why are you seeing an upregulation in mitochondrial genes if the primary route of action is TET enzyme interaction? Is there some sort of crosstalk between nuclear methylation and mitochondrial function?
Yes, I think there is a crosstalk, definitely, because we can see the genes that are involved in mitochondrial, transcription. They are also showing similar pattern in our database. I was not able to show over here, but also we looked at the mitochondrial genome methylation in fish. They are all gene body methylation, and they are very little, 2% methylation. Yeah. But when we looked at the human cells, we did WGBS on human cells after vitamin C treatment. The mitochondrial genes come back. The expression of these mitochondria, we did RNA-seq as well as WGBS on those in vitro cell culture as well. So we found out the mitochondrial genes actually, expression come back with vitamin C.
Right, but yeah, without any methylation, then I'm just wondering what the mechanism would be.
I think transcription of the mitochondrial genes are not probably regulated by methylation. Yeah.
So following up on the mitochondria, I mean, Mike Hudeman, if he's on Zoom, he would, I mean, how healthy are the mitochondria? Maybe the vitamin C is just restoring their polarity and that's allowing expression to occur. So vitamin C is correcting the defects of the mitochondria, not.
Probably, I agree with you, right? Probably it is, it is antioxidant. My vitamin C not only works as epigenetic modifier, it is also a very strong, kind of antioxidant. And I think it's working from both, corner, that acting as, antioxidant, also acting as epigenetic modifier, or it is changing that the same way that.
Did you look at mitochondrial polarity or anything?
No, we have. I think this is to do in the future. Now, my new PhD student joining this August is going to look at aging and mitochondrial polarity, mitochondrial membrane potential, and also the other. I don't know what he said. Something cool.
And you can just get molecular probe dyes that, stain the mitochondria. It tells you how healthy they are with properly polarized. My other question was about how are these transmitted in the sperm? Did you look, I mean, you said sperm epigenome, but the slide said fin epigenome. So did you actually look at the DNA methylation in the sperm?
Oh, sorry, I should have said fin, not the
Yeah,
sperm,
yeah.
actually. I mean, that was my tongue slip, sorry.
But are you looking at the sperm also?
Yeah, we look at the sperm, but we did not check the mitochondrial genome. Yeah, we checked mitochondrial genome in the sperm as well, but I don't know exactly what the answer
You
is.
see DMRs in the sperm because you're from Skinner's lab and he looks at a lot of
Yeah.
DNA methylation.
Yeah.
Do you see any DMRs in the sperm or?
Yeah, most of the data that we presented from F0, F1, they are sperm.
They are sperm.
Sperm, yeah. They were sperm, and only the somatic cells we looked is the F2 generation somatic cell. After the germline transfer into the somatic cell, they are supposed to be erased because of reprogramming of the somatic cells, but they were not.
So they're actual sperm, not the testes. I mean, you get the free swimming.
Yeah. There's free sperm, yeah. We actually isolate the sperm and then get rid of those tails and only the sperm head, and yeah. And density centrifugation, we actually remove them both mechanically and also biochemically.
Excellent talk. I have two questions.
Mhm.
In the PCA plots that you showed for ovary and liver, did you mean that taking a swab of the fin was able to
No,
differ-
not the fin.
Oh, okay, but like it was not the ovary or liver tissue that
No,
gave
we,
it.
we, no, we did not.
Okay.
So what happened is, we sequenced ovary and liver both, right? But when we checked the match between these, we were able to see the pin can actually predict. Yeah. Pin can predict the phenotype and the changes in the ovary and.
Second question was that because you spoke about having some human cells and you could see the fat decrease in the vitamin C treatment. Do you have any data on like human semen samples with and without vitamins, like from men who take with, okay,
Rick.
I'm Rick's student.
Okay. Yeah, you will have. We just talked. Yeah, this morning. Yeah, I think it will be very cool if you can go and ask. Right now, the one good thing is, our urology department at Mizzou, has started recommending vitamin C to their patients. And then they have agreed to give us a sperm sample. These are normal human beings. We just wanted to see whether the sperm gets better, right? The sperm epigenome gets better because of the vitamin C treatment or no. We have like 21 patients we have agreed so far, but 21 will be enough for me. Right? So this will be probably 5-6 months from now. Yeah, we'll be able to see just the basic. And another thing I want to say is in normal medaka fish, we actually treated them with vitamin C quite early in development as well as in adulthood. Normal medaka fish have very strong, very nice looking sperm morphology, as well as the histology is beautiful of the testis. So it means naturally, I think it is also helping the testis, to grow better. We have that from our recent experiment. So this is good as well.
I have a question from our chat
Yes.
box on the Zoom feed. This is from Dr. Candy Ding.
Mhm.
He has one question regarding the offspring phenotype. It is interesting to see that the spontaneous pathology caused by prenatal exposure to these toxicants. And in some other cases, the phenotype is shown only upon a second challenge postnatally, such as viral infection or carcinogen exposure. Do you think epigenetic modification play a different role between these spontaneous phenotypes and stimulated phenotypes?
Yes. So yeah, that's a very cool question. One of my graduate students just finished the thesis this year, and he did one with the immune challenge against those, F4 generation individuals that have the history of exposure or no history of exposure. When he challenged against LPS and poly-IC, he saw the immune, function was compromised in the fish with a history of BPA exposure due to the second hit. And another student challenged against PFAS, And she actually saw increased fertility defect in the males. And in the females, not too much defect, but the liver disease was severely, what can I say? Liver was severely affected. There was already damage in the treated individuals, but the fat accumulation had gone even further in 100% of them, suggesting that these offspring with previous generations exposure or prior exposure are probably more susceptible to the chemicals of today or tomorrow. Is that the question?
I believe so.
Oh, okay, I think I thought so.
So for the fertility effects, I mean, if they have PCOS and male fertility, how do you actually do this experiment? How do you breed them? Because the medaka have a small clutch of eggs stuck to their bodies. I mean, how do you get enough offspring to go to the F4 and beyond?
Yeah, we actually do is we collect the eggs. We need only 50 to 75 eggs, and then each individual produces 20 to 30, and we pool them from 3 days and put them separately in different. Today's will be in one tank, tomorrow's will be another tank, and the after tomorrow's will be another tank. We just raise them.
If you get enough fertile
Yeah.
embryos that way, I mean, what is the decrease in?
Oh yeah, so 25% decrease in fertility.
Okay, so
Yeah.
there's still enough to
Yeah.
do the experiment.
Do you have any people with a psychology background on your team?
No.
I'm hoping this is not going to be a trite point then, but you mentioned a change in their sexual behavior. So you're not just looking at fish that are generations after an exposure to a chemical, they're also exhibited a different behavior. So aren't you even a little bit curious to dig in to find out why the sexual behavior has changed? Is it that they can't see as well, so they can't tell what's a female and what's a male? Or is this of no interest to you?
We have not done that, but when we do back cross, right? So with the opposite sex of the, male from the control lineage and the female from the treated lineage, that male is trying to mate, the female does not mate. Or, when we do opposite, then the treated line is not interested in the beginning. So then there is a wait time of like 2 to 3 hours, sometimes 5 hours for mating, even with back crossing. So suggesting that individuals from the treated lines have some sort of, sexual dimorphic programming of the brain. That's what my neuroscience faculty said.
Yeah. My background is neuroscience, so
Yeah.
I think it's an interesting question.
Yeah, it is. And the last time I presented this to Rutgers P30 program in September. And I think Lauren, probably you may know Lauren over there. So she said, it is very kind of interesting, but I need to be probably more specific about when I use the terminology. And I started not talking about this. It could be like, translated differently by public. So I talked to my experts in our department and also the, medical school about sorting this out. But we have 10th generation going on, and they have severe, problem of mating as well. We don't get any fertilization anymore. And they are dying within prematurely by 9 months or 8 months. They are already, aged. Acceleration of aging is enhanced by 50%. Their lifespan is like 2 years. They die, prematurely. They show scoliosis like, symptom by 6 months. And they cannot mate after their body is bent. And then they do not fertilize the egg either. And we have seen this, and we're not going any further than 10th generation now.
I guess the follow-up question then is, have you tried a related exposure to see whether you get the same effect on the behavior and in the generational effects? Or are you just sticking with BPA for the rest of it?
Right now, we are sticking with BPA, but, we had the line from atrazine exposure. But the atrazine exposure went up to F3 generation, and we saw some behavioral problems with atrazine. But we didn't have a recording system at that time, and we just recorded in our cell phone. But that was not enough to show as an evidence. And then we also tried with ethanol estradiol up to F4 generation, but we didn't pay attention to that was little early on to any behavior. After that, we just followed up with BPA, no more other. Oh, THC. We actually did THC exposure and up to F2 generation only, and THC had a distinct behavioral change in the F2. They were more addictive to either eating or addictive to do something like the same thing every time.
DSE is,
THC,
oh, okay.
a THC like delta 9 THC from marijuana, cannabinoid. And F2 generation was like behaviorally completely messed up. Yeah. And that paper is now under review, but we are being hit by people because how did you use THC without doing air water interface or something like that? And we are having trouble publishing the paper because we put that in water and dipped our fish in the water. Yeah. So we're trying.
Yeah, I'm sure you'll find the right journal.
Yeah.
So just thinking about BPA, so most fish systems are made out of polycarbonate. So I guess my question is your fish system made out of polycarbonate? And can you measure BPA in your control animals? You can.
We did.
Okay.
We measure and we have glass tanks. Yeah, all glass tanks. So wherever we do the experiment, they are actually raised in glass petri dishes. They are like kept when they are juvenile in glass tanks, and they are transferred to glass tanks. And we measure BPA in water. Yeah, and we actually outsource to what is that company, Eurofin? Yeah. The latest, we found some BPA in our filtered water. So milk filtered water had BPA in it. We send that as a reference, with no BPA. It turned out we have the same amount of BPA as the, almost tap water has. So it means, the membrane they use for filtration has BPA in it.
Yeah, Scott Belcher reported this back in the day.
Oh, okay.
Yeah, that BPA leaches out of the milk water systems, yeah.
And we have reverse osmosis right now, and it goes from that Culligan system, and then filtration. And still those filters are loaded with chemicals. That's why I said, we cannot get rid of these chemicals. We need to find a way to, correct it by ourselves. Any more questions?
Maybe one last
Okay,
quick one,
okay,
just to be
yeah.
done. Can you prevent against? So you've shown that the vitamin C can help ameliorate a phenotype that exists. Can you prevent against the phenotype before it develops?
Yes.
Okay.
We did the experiment, 2 experiments. One experiment with co-exposure during early development, vitamin C alone, vitamin C plus BPA, those 0 to 15 days, and then BPA alone and control, right? So we did that experiment, and we were able to prevent completely because, the early on, the BPA effect did not go through. And we saw better, in the liver, we didn't see as much as expected, but however, in the reproduction part of it, we saw a really massive, response, betterment, I can say improved fertility, as well as the histological feature of the gonad. And the another thing we did is we actually treated the adults, and then we co-treated with vitamin C, those adults, to see whether they produce the offspring with defect. And we also checked their fertility. So the vitamin C is able to reduce the, infertility issue, but cannot overcome completely in the normal adults if they are exposed as adults. Probably they need early stage programming to probably take effect. However, if we use, for example, F2 generation adults that have the prior history of exposure, and then we treat them with vitamin C, only male, we have not tested females, only males, the F3 generation does not have NAFLD. So it means the F3 generation offspring do not develop liver disease. That histologically we can confirm. But however, it might be going from the maternal part of it as well, because we had experiment of male line, female line, male and female together. Since both contributed to disease, then we started mixing them. Because male line also contributed to liver disease, female line also contributed to the same female liver disease. And for fertility, male line is responsible, no female line. So it's a maternal transfer, probably it is not of maternal origin, it's a paternal origin. So interesting, but yeah, there is a way to prevent, to answer your question. Now we started with PFAS. And PFAS is more like stronger than BPA in terms of producing these phenotypes. And we're testing different concentration of PFAS and the ability of vitamin C to correct every one of them. And we started with micro-injecting PFAS into the embryo to see whether early embryonic reprogramming can also be corrected by the PFAS induced, So we injected with vitamin, 10 nanoliter, 1 micromolar vitamin C. And, we don't have the data yet, but we did that experiment. And in vitro, we can see in Sertoli cell line, Leydig cell line, and then germ cell line, the vitamin C can act on Sertoli cell, but less in germ cells, okay, directly in the differentiated germ cells. And then the undifferentiated spermatogonia, those cells respond to vitamin C, not vitamin E. So they have selective response. So suggesting that maybe there are mechanisms how these vitamins are working at the epigenetic, signaling pathway, and these pathways are at different stages in different stages of cells. So vitamin C is really good for liver. That's I can say. Really helpful for liver health. And for others, probably these are all secondary mechanisms acting, like PCOS phenotype was found to be corrected because liver was corrected. And we believe that microbiota are also corrected because intestine was developing much better than the untreated ones. So there might be a secondary, hit by the product of those pathways to, maintain the health. It means like my speculation is if you initiate a pathway, then pathway will take its own way. It will correct everything one after another. But you need to give a right direction to that pathway. You don't have to correct all 100, 2,000, 20,000 genes. You may want to correct only the main highway, and it will take you to the destination. That's what I feel. But this is speculation, not scientifically proven, please. Yeah.
Well, let's thank Ramji again.
Yeah, thank you so much for making.
Let's see. Do you want me to bring him down to your office or he comes to?
With Graham.
Oh, you did. You want me to bring him to your office or?